is outside the scope of this event, so you don't need to bother with those questions. Feel free to line up at the microphones, and while we're waiting for people to line up here, we'll take our first online question, please.
Thank you, Jeff. What is your recommendation to handle significant peptide degradation and dissolution method in vitro method to calculate percent release? Meaning that, what is your recommendation to handle the significant peptide degradation and dissolution method in vitro method to calculate percent release? Meaning that, do we need to add degradants or only active to calculate the percent release?
I'm not sure I understand the full question there. You're talking, the question is asking...
Can you give it to us one more time with a little bit more oomph, and can I get more energy on that mic, please?
What is your recommendation to handle significant peptide degradation and dissolution method, in vitro method to calculate percent release? Meaning that, what is your recommendation to handle significant peptide degradation and dissolution method, in vitro method to calculate percent release? Meaning that, do we need to add degradants or only active to calculate the percent release?
I think I understand the question. I'm not sure what sort of formulation you would require a peptide to undergo a dissolution study, but so I'm not sure how to answer that question.
Yeah, that could be in an extended release, so it's a little slightly outside the current discussion. But on the other hand, I think the people raised the question is if there is, during the dissolution method itself, the API have significant degradant. So how that should be handled, whether it can use a control to, you know, sort of as a baseline to correct it or not, I think maybe comment a little bit on that aspect.
Yeah, I think we'd probably have to ask the firm to provide question to the agency via correspondence or pre-ANDA mechanism.
All right, let's move on to microphone number one. Nice and close for me, please.
Hello. Thanks for wonderful presentation, all web presentation. I have two questions related to the peptide characterization. One, in your presentation, it is many slide I have observed you have mentioned significant number of lots to be used for characterization. So in FDA's view, what is significant number? That is first thing. And also you have mentioned that characterization should be done near expiry as well as at the release. So the main problem as a development company faces is to getting a near expiry RLD. And in that situation, is it acceptable to buy an RLD, put it under accelerated condition and use that as a near expiry as an approach?
So for the first question regarding the number of batches, significant means at least 3. However, it's in your best interest to characterize as many batches as possible, because especially for the RLD, that will allow you to better characterize the variability in these various attributes. Regarding the second question, just quickly remind me what that was.
You mentioned that we should use the near expiry RLD
that's right,
for characterization. So whether buying
okay.
RLD and using it, putting it under accelerated condition and use that as a near expiry, because we may not get it in market near expiry RLD, 3 lots especially.
We would not accept RLD aged under accelerated conditions. For that type of study, you have to obtain RLD that's been aged near to the expiry date.
Thank you.
Okay, and now some of you may be under the impression that since it's after lunch, the threat of having to buy lunch for everybody doesn't apply anymore if you try to sneak in 2 questions. But there is a networking event coming up. If anybody else tries to sneak in 2 questions, you get to buy the first round of drinks at the networking event. Microphone number 2, please.
Another question for Dr. Smith, and this is kind of related to the previous question. This is for the drug product comparability studies and the requirement for the proposed product to run the studies at or near the proposed expiry. So if a firm had a proposed expiry of, say, 18 or 24 months, one would have to wait until we acquired that data before submitting the ANDA. Would this preclude the possibility of a 10-month approval? Typically, we would submit with at the 6 month, right after 6 months of stability.
So yes, that would not necessarily be a requirement for submission, but for approval of the ANDA, it would be a requirement to provide that end of shelf life data.
So the new shelf life data could be provided during the review process?
It could be,
The
yes.
timing, okay.
I mean, I think part of the reason we're communicating this information is to try and help you design the logistics of your program and time your submission appropriately to optimize the review of your application. So if you could provide that data on submission, it would probably facilitate the review.
Thank you.
Okay, let's go to an online question, please.
Could you suggest an orthogonal technique to SEC HPLC for determining the aggregation potential of a 37 amino acid peptide drug?
So much for specific questions.
Yeah, that's quite a specific question. Probably be quite difficult to answer without further information.
And also a reminder from this morning, if you have something that's very, very specific to your situation and or application that will not be a general learning opportunity for everyone involved for the event, try to hold those for perhaps a different venue. Microphone number 1, please.
So my question is related to Q3 similarity, for example, like for PLGA products, molecular weight and polydispersity. Do the methods need to be validated, which are used for showing similarity between the test product and the RLD?
Sorry, can you clarify, are you talking about like an API or are you talking about an excipient like polymer for your polymolecular weight and PDI? What your
So it's all kind of characterization. So characterization between the API similarity as well as similarity of PLGA, such as molecular weight of PLGA and polydispersity.
So I think on your second question, it might be good to hold off. We're going to have a whole session after this about the PLJ specific and the inactives or the excipients. But in terms of for the active ingredient, I might hand over to one of my other colleagues to kind of answer on if that needs to be validated or not.
Go ahead and repeat your question again about the active ingredient.
Sure. So, for example, you had shown in your slides for API similarity, there are methods like XRD and NMR. Those need to be validated methods?
Generally, if the method is a quantification type method, then you need to validate. Yeah. If it's a qualitative method, then no.
All right, microphone number 2, please.
Good afternoon. I have a question for Dr. Smith. In your presentation about container closure system, you mentioned the need to demonstrate comparability of leachables between test reference products. It might not be possible. My question to you is that if we demonstrate a test product gave lower levels of leachables than the reference, is that acceptable with the agency?
First, you should be striving to reduce the amount of leachables as much as possible in your proposed product. That's really the first cutoff. If you can't do that, that's when we ask for a comparison with the reference product. So I'm not sure what the last part of your question was.
So it's okay to be better than the reference in terms of leachable profiles?
Oh, yeah, of course, yes. Yeah, of course.
Thank you.
Sorry.
Online question, please.
Should the comparability studies be conducted in a GMP laboratory?
No, that's not required.
See, those are the kind of online questions I like. Microphone number 1, please.
Clark.
Yeah, please, go ahead.
The batch that manufactured that I use for manufacture of the material for the test obviously needs to be manufactured under CGMP. But the laboratories, no.
All right, now the microphone number 1.
For a complex product that's a colloidal suspension of varying molecular weights, how can a company control for possible formation of dimers, trimers, oligomers that would tend to confound the results and is not always repeatable? Is there some way to control for that?
So I think in terms of, you're asking about the optimization of your analytical method to make sure that you're not getting an aggregation that would be different to what your actual drug product would be when you're actually making it, or?
There's not another question. No, it might happen. Some iron colloids form dimers, trimers in solution on aging. How do you control for that? Because that would tend to confound the particle size distribution.
I think that's quite good, and I might hand over to Xiaoming, but prior to that, you can do a lot within your method development. You can use things like the mother liquor and things like that. Let's say if it's in a soluble, like there's usually a carbohydrate or something like that, a certain concentration, rather than using something like a diluent, like just DI water or something that may cause a change to the formulation. I would use like the mother liquor or something as close to that as possible, and then use that as your diluent, and then doing the justification that you use this because of these reasons, and even put some of the control, the diluent, to show the fact that it does aggregate into those conditions and help to justify that. I think an awful lot of this is what we're asking for is the scientific rationale and the background information really to show that your method is appropriate and your testing method is appropriate for what you're developing.
Thanks.
Okay, and I'm trying to keep track of who came up when. I think that you were next in line if you want to step up. Now, you were up before this microphone had anybody at it, so you get to go first.
Hi. There were a couple of references to the synthetic peptide guidance in the presentation. Is it accurate to say that that guidance establishes the filing criteria and then the complex product guidance has established the review criteria?
Your question was about the complex peptide guidance being what criteria?
Does that establish the filing criteria, the eligibility for the ANDA pathway, and then the additional guidance is concerning complex products? Do those supply the review criteria, or is the peptide guidance a review document? Are those the standards you apply to on approval?
Those factors described in the guidance are for approval, not for filing.
Okay.
Yeah. So you need to satisfy those factors to have your product approved by the 5J pathway.
But if a product satisfies those factors and is filed, does that mean the product meets approval criteria?
The review of those factors doesn't get performed at the filing stage.
Okay. All right. It's just the peptide guidance says that it's a filing guidance.
Okay. Well, the filing reviewers won't review that material. That'll be reviewed later by CMC reviewers.
Okay. Thanks.
Online question, please.
This is actually referring to Dr. Zhang in slide 16, the calcitonin salmon impurity analysis. 13 nasal spray drug products were analyzed, and out of the 100 impurities seen, 4 were above the 0.5%, and 16 were above 0.1%. Were these surveillance samples of distributed products, and if so, did they match the ANDA release profile and specifications?
It's like Quiz Bowl.
No, for us, we basically just try to develop a analytical approach to analyze those peptide impurities. So it's more like a method development. So when we study those samples, some of those expired samples, the one that had the most impurity are those from the recombinant DNA peptide. So it's not simple saying like, whether it's release criteria or something.
Okay, microphone number 2, please.
In the first presentation, it is mentioned that identify and characterized above certain threshold. So what should be the identification threshold for peptide impurities for formulation and API?
So the identification threshold is not something that's described in any guidance at this time. The peptide guidance that's been published, you're asked to identify all peptide-related impurities that are above 0.10% of the drug substance. That doesn't necessarily mean that in the end that will be your control threshold. However, you will need to be able to control those impurities that are detected above that level, whether they're controlled as unidentified impurities or not. So for peptides that aren't covered by the guidance, you can propose whatever IT you feel is appropriate, and that will be a review issue.
Microphone number 1.
Hi, this is related to the particle size 3-tier specification requirement for a material API, which is ultra micronized and no impact on the CQAs of the drug product of using the micronized material, which is a D90 is around like less than 10 microns. So when such a low micronized material and not much impact on that one, is a single specification, like a single tier, like a D90 spec is adequate, or still we need to go like a D10, D50, and D90?
The spec, I think that's a review question, so I would defer that to the actual review to decide whether a single tier or multi-tier is needed. However, for characterizing the particle size that you mentioned, suspension less than 10 micron or API dispersed, and having a single tier may not be enough to give you information about the polydispersity or how it can change. So from a technical perspective, I would think you need a multi-tier, D10, D50, D90, in order to give you entire picture about the polydispersity of the API, where it is, and whether it is bimodal or monomodal distribution. So those information with the 3-tiered numbers can give a better information. And again, the detailed measurement condition has to be included in order for correct interpretation of the D10, D50, D90. And depending on the instrument, those numbers can mean totally different things.
I agree. Thanks.
Okay, microphone number 2, please.
Okay, thank you. Actually, I don't have a question, but I have a clarification about the early online questions about GMP requirement for, I guess, for method validations, basically for method validation requirement during the product development stage. So I think we have a panelist tomorrow morning from 9:30 to 10:10 from OPF, OPQ, and he probably can better address this question. I just don't want to misleading and give any wrong information. Hope that person asks questions still there, so he can come tomorrow morning.
Thank you for that. So whoever posted that question online, tune back in tomorrow morning. The panel will be able to address that then. Speaking of online questions, let's go ahead and take one of those.
This question is related to the characterization of the particle size of a suspension product. If we have concern about the existence of interference of formulation ingredients on the results, such as presented by the speaker, what is the best way to approach this with FDA?
Certainly, you need to evaluate and do enough homework to understand whether you do see interference or not. Because if you have concern there's interference, and if you have sufficient data to support there's interference, I think that's a legitimate scientific question to discuss. So I think the proper format probably would be pre-enda.
Yeah, I think, as Xiaoming said, is if you've got enough information to justify that that's an interfering or an artifact of the analytical method and you want to discuss that in more detail, that can come through either a specific control correspondence or more likely through a pre-end a meeting where you can actually present the rationale and all the background towards it to present that full case if you want to present that to the agency. Kind of explain what's going on.
Okay, microphone number 1, nice and close for me, please.
Thank you. My question is related to the setting the limits for the peptide impurities. So the question is, do you consider to develop specific guidelines for setting the impurity limits for peptide products?
This is quite a common question that we get regarding peptide impurity limits. So there's no guidance currently, obviously, for that purpose, and we don't have anything in preparation at this time. So you're really left with supporting your proposed impurity limits based upon comparison with the RLD and justification, in absence of that data, justification via toxicological testing, or if there's immunogenicity risk, testing along those lines as well.
Microphone number 2, please.
Thanks for the presentation. I have a question about the particle size, because we have worked with suspension formulations, and we did find, like Dr. Xu mentioned, like the particle size were affected by the addition of the polymer, the hydration, and so we will work with like using different, and also dilution. So we work with different medium, dilute them to measure the size. But I was wondering, like, what kind of data, like, we need to provide to convince reviewer, or like, if you have any more detailed guidance for this topic.
Sorry, can you repeat the last bit? So you said you're seeing for a suspension product that do you see there's
yeah, the size, the particle size were affected by the
dilution.
addition of the polymers.
Polymers.
And also the dilution will also affect the particle size measurement. So like in this case, like what kind of data we need to provide to convince the reviewer?
I think that depends on one, why is the measurement of particle size important? So if the particle size is relevant for the dissolution, or is relevant for the bioavailability, or relevant for the bioequivalence determination, so you need to have other means to give you a different measure of whether the presence of interference going to change the outcome of that determination. So if it does, for example, if diluting, you do notice a size change, and or presence of excipient, you do think is causing the size value to be different. Again, I think the more information you can provide, the more justification, the rationale for the selection of the material and the procedure, and those will help the reviewer when they are analyzing it, so they can give more objective assessment. And granted, there will be challenges, so that we do recognize. And the more you can provide, the more detailed that justification you can provide will give a better, will allow the reviewer to better assess it.
So I've also got a little bit of a follow-up on that one too. I think one of the advantages when you're developing a generic is you always got that nice standard, which is the reference product. So if you actually, and we're expecting that the reference product should act in a very similar fashion as your product. So if you're showing the fact that your particle size may be changing of your product with that dilution or similar conditions, making that measurement of the reference product and showing the similar sort of effect helps actually gain sort of support that your product is acting in a very similar fashion, has the same physical chemical properties, and therefore should be relatively equivalent to that reference product. So I think it can be both a dilemma from your perspective, but also can be helpful to this other aspect, is it's building a better sort of case. So as Xiaoming was saying there is, there's a lot of things that you can do in terms of data for developing and optimizing the method and showing that you have this change within the particle size and rationale as to why it's changing, but you always have that reference standard to measure against, showing it's having a very similar effect. And I think that helps prove the sameness of those two products to a certain degree. I don't necessarily it's always the best way, but it actually does can be supportive for your development program and as well as evidence.
Thank you.
Online question, please.
Is a patient toxicity study required to evaluate a leachable component?
Unnecessarily.
Okay, we'll leave it at that. Online people, tune in again tomorrow and ask for a bit more detail. Microphone number 1, please.
So I have again 2 questions for particle size. One is if we are using during the manufacturing FVRM as a method as a PAT tool, and finished product is measured by different method. So the question is, do we need to validate the in-process particle size method using where we are using PAT tool? That is the first question. And second,
No, you don't get a second one.
Okay.
No. Okay. Stop right there. Go ahead and answer that question. We got other people in line. Go ahead.
Okay, thank you.
Well, so you're asking for the in-process control particle size measurement using a process analytical technology, PAT. And again, I think that's a review question. So I'm from the lab. I would try to stay away from what would be determined appropriate, not appropriate. But as any analytical method, if you want to show that you have an understanding of what you are doing, the validation is just to show that how you understand it. It's not to follow a specific numbers or let test you to do, but to show how do you understand. So along those lines, I would think you do need to show why that particular technique is suited for in-process monitoring. It's challenging as is already for finished product characterization, and if you want to use it for in-process, that'll be more challenging. So,
the background to it, like in process, it's a suspension.
You can pass that during the networking event when you're buying the drinks.
Thank you.
Microphone number 2, please.
Good afternoon. Thank you for the wonderful presentation. This is regarding the higher order structures by NMR. Just a comparison with respect to RLD is sufficient for the submission or for the approval of an ANDA, or should we also calculate the percentage of the higher order?
I didn't quite get that last part of the question.
Could you repeat
regarding
the
the
last part?
percentage?
Do we also have to calculate the percentage of the higher order structures, or just a comparison against the RLD is considered acceptable?
Okay, so you're referring to different types of secondary structure, for example, alpha helix, beta sheet, for instance. So we see those kinds of calculations provided for both CD and FTIR. For NMR, we're more interested in the overall three-dimensional structure of the compound in solution. So those types of analysis usually aren't presented. What we normally see is more of a quantitative comparison of the structures in solution. There are a number of agency publications that I can refer you to if you come ask me later on. I can give you those references.
Okay. And I apologize to everybody who's still got questions. This was obviously a very high interest topic, but for the sake of keeping us on schedule, we're going to move on. And we're going to move on by thanking our panel for coming and sharing this with me. Panel, you're free to go. I do ask you to try to make it out in the hallway before you get swarmed by people, just so that we don't block some of the aisles right here. We are going to take a 20-minute break. It is 3:10 now. We'll be starting back up at 3:30 with our last session topic for the day.